Bismillahir Rahmanir Rahim. Alhamdulillahi Rabbil Alameen. Wassalatu wassalamu ala Sayyidil Mursaleen wa ala alihi wa ashabihi ajma'in. Okay, so inshallah today we're going to continue from where we left off yesterday and develop on the scientific arguments for and against evolution. And remember, there's the two parts of evolution. The first part regarding universal common descent or universal common ancestry, that all living things are related to each other by ancestry. And the second part, which is that transformations can happen from one life form to another, from one type of living thing to another, and novelties, innovations, new characteristics, and new features can arise in life by natural processes. So we're going to deal with the first part of evolution to start with, and then move on to the second part. Right, so we're going to start with the fossils. And normally people who are not well read about science, or who are not scientists, this is what really confuses them. So fossils are the remains or the imprints, the impressions that are left by dead living things within rocks. And rocks are formed in layers. So the rocks that are lower down signify an earlier period, and the rocks that are higher up signify a period that's more recent. Now, in terms of Darwin's theory, paleontologists are the specialists in fossils, were strongly opposed to Darwin. And probably the most knowledgeable paleontologist of his time, Louis Agassiz, who we encountered before, he opposed him, and he opposed him in writing. And we're going to mention some of his arguments, which are still relevant, surprisingly, till today. So now, taking a very superficial reading of the fossils, one could be mistaken or led to believe that they suggest universal common descent. Right? Because what you find is in the lower rocks, the fossils that you find in them are simpler, are more primitive and less complex animals, or less complex living things. And then as you go higher up, they become more complex. And the ones that are lower down are less like the animals that we see today. And then as you go higher up, and you see the more recent fossils, they look more like the fossils, or more like the animals that we see today. Right? So Darwin and his followers would argue, based on that the animals in the later period are descended from the animals of the earlier period. But as I mentioned, Louis Agassiz, what he said was that there's nothing like parental descent between the animals of the earlier period and the animals of the later period. And there are very good reasons, very good scientific reasons to say that. And So if we were to say that it's not universal common descent that explains this pattern, then what is it? So as I said, I quoted Agassiz yesterday, where he mentioned that this is a progression, a logical progression that we should attribute to the mind of the creator. So the creator created living things according to a certain pattern, and then eventually leading up to the living things that we, or the types of living things that we see today. And that's not just an assertion, but it's what you get from a literal reading of the fossils. Remember, Darwin and his followers don't believe, or the majority of them don't believe in the literal reading of the fossils. We'll come to that shortly. Now there are three features of the fossil evidence, of the fossil record, that go against evolution, that go against universal common descent. And the first is, right, they are found anywhere in the world, and also from the same ground. So you have certain famous sites where fossils are very well preserved, and those layers are dated based on some dating methods that they have, right? So you probably heard of the Cambrian. So the Cambrian is actually a place in Wales. So and that's where the first fossils within those layers of rocks were found, and they saw this huge burst of animal life in that layer of rock. And then they found that within that same period, within those same within rocks dated to that same period, in different parts of the world, they found the same burst in animal life. And that was, and you find that in this famous fossil site called the Burgess Shales in Canada, and then you have a site in China called the Xinjiang, and you have this is something that they with regards to what you ask, if it's from the same ground, maybe then they could be descended. That's probably something we can come back to, inshallah, if you can ask that towards the end. This is something that was accepted from Darwin's day, which is that new forms of life, when they appear suddenly, without any connection to previous forms of life. So it's not like you have one type of life that is slowly, gradually leading up to a new form of life. Rather, when you have one form of life, it just appears suddenly, fully formed. And the same with the major features that we are familiar with. Like when eyes appear, they appear suddenly. It's not that you have a quarter eye or a half eye or a three-quarter eye, and then you have the full eye. No, when the eye first appears, it appears suddenly. And same with the wings, same with teeth. So it's that you don't see a gradual transformation of one type into another. And it's the same with human fossils, as we will see shortly. So this is just an example. With regards to turtles and tortoises. So the turtle and the tortoise is a very unique animal. It's a reptile, so it's a vertebrate. It has a backbone, and uniquely amongst vertebrates, it has this shell that is made up of bony parts called scutes, and it's about 50 different bony parts. And its shoulder bone, the scapula, so again, uniquely amongst vertebrates, is positioned under the ribs and under the shell. So this is a complete transformation. But when the turtle appears, when the tortoise appears, he appears suddenly. And he appears in rocks that are dated to about 200 million years ago. Right? So this is for a quote from a paper. The turtle's shell represents a classic evolutionary problem, the appearance of a major structural adaptation, and they refer to this instantaneous appearance of this evolutionary novelty. And this is a mainstream paper. And this is not unique to the turtle shell. This is typical. This is the norm. This is a quote from a textbook from the mid 20th century. So most major groups of animals appear abruptly in the fossil record, fully formed and with no fossils yet discovered that form a transition from their parent group. So obviously they assume that there is a transition, but there is nothing that is there in the fossils themselves that would suggest that there is a transition. So that's just assumed. Now one of the most remarkable examples of this is the Cambrian explosion, which is sometimes referred to as evolution's Big Bang, or the biology's Big Bang, where you get a huge burst of animal life in rocks that are dated approximately 500 million years ago, and they appear suddenly. So the first vertebrate fish appear in the Cambrian. And you don't have complex animal life before that. And there's nothing before the Cambrian, what's called Precambrian, clearly leading up to what you find in the Cambrian. So as an example, when you talk about eyes, there are different types of eyes, but there's two types of eyes which are the most complex. One is the vertebrate eye. The vertebrate eye, meaning that vertebrates have, like birds and mammals, and so on. So the vertebrate eye is called the camera eye, or the simple eye. So what you have is an eye with one lens, which focuses the light onto light sensing cells, and then that's transmitted via nerve cells to the brain. So that's the camera eye. And then you have a different type of eye called the complex eye. Sorry, not the complex eye, the compound eye. And you have this in insects and arachnids and other types of invertebrate animals. And with them, what you have is not just one lens, but many lenses, so hundreds. You have lots of units, and each one has its own lens and its own set of light sensing cells. So these are the two most complex types of eyes. And they are both created, or they both appear in the Cambrian without any evidence of simpler eyes or any eyes at all before the Cambrian. So there's a quote here from a 2013 science paper, a mainstream paper. It says, the available fossil record illustrates that the Cambrian explosion spawned the simultaneous birth of the principal invertebrate compound eye and the vertebrate camera-style eye. Right? So the most complex eyes appear right at the start of complex animal life. And this is important because we'll see examples of this, probably a couple more times, but there's many examples of this. This is an example of where the theory clashes with the evidence. So you see, Richard Dawkins, for example, he hypothesized how, according to Darwinian theory, eyes might have formed. So he said that you could have had, a half eye or a quarter eye, and that slowly led up to a full eye. And he points to some examples of simpler animals who do have simpler eyes, like some jellyfish have simpler eyes. But the evidence shows that the most complex eyes comes before or comes at the same time as the simpler eyes. And it's pure speculation. There's no demonstration of how you would get from a simpler eye to a more complex eye. If anything, we see that those animals with simpler eyes, they've just stayed the same throughout their existence in the fossil record, like jellyfish. And as I said, this is the norm. This is a quote from Jeffrey Schwartz from a book that he wrote in the 1990s. He's a scientist, a prominent scientist from America. He said, "We are still in the dark about the origin of most major groups of organisms. They appear in the fossil record as Athena did from the head of Zeus, full-blown and raring to go, in contradiction to Darwin's depiction of evolution as resulting from the gradual accumulation of countless infinitesimally minute variations." So you will actually find scientists who oppose the Darwinian view of gradual transformation while still maintaining a materialist view of the evolution of life. So Jeffrey Schwartz is one of them. He's a materialist when it comes to life and the evolution of life. But he proposes other possibilities that which fall under a set of theories called the extended evolutionary synthesis. And in reality, those also suffer from many of these similar types of weaknesses. As I said, this is the norm and not the exception. There's a contemporary paleontologist by the name of Gunther Beckley from Germany, and he's written an article where he basically collects all the research papers and all the written works of paleontologists from who specialize in different periods, showing explosions and radiations and bursts everywhere. So you have things that just appear suddenly in the fossil record without any connection to animals that were there before. So what is their answer? What is the answer of Darwinians? Their answer is that the fossil record is incomplete. So the majority of the animals do not fossilize. Right? So the majority of the animals don't fossilize. And that's true. So he says that the transitions from one type to another, the reason why we don't see them is because they didn't become fossils. Darwin said, "I look at the natural geological record, meaning the fossil record, as a history of the world imperfectly kept. On this view, the difficulties above discussed are greatly diminished or even disappear." It's the same argument that's presented by Richard Dawkins and Darwin's contemporary followers. And this is a response by Louis Agassiz. He has an article called Evolution and Permanence of Type. It's an essay that he wrote against Darwin. And Darwin was not a fossil expert. Agassiz was. And he firstly says that this is it's a negative argument, as in you're making your theory unfalsifiable. Regardless of whatever evidence we present, you will just say, "Oh, that doesn't agree with the theory because there's not enough, because there could be other evidence that are found, that will be found." So you could do that with anything. So he says, both with Darwin and his followers, a great part of the argument is purely negative, thus they throw off the burden of proof. And he says, however broken the geological record may be, there is a complete sequence in many parts of it, from which the character of the succession may be ascertained. And he says, you get you get fossils of animals and of even the embryonic phases of animals, meaning while they're going through development, preserved in the rocks. And they're called so animals without bones are preserved. So he says, since the most exquisitely delicate structures, as well as embryonic phases of growth of the most perishable nature, have been preserved from the very early deposits, we have no right to infer the disappearance of types because their absence disproves some favorite theory, meaning some favored theory. So just because this doesn't agree with your theory, you can't just postulate that therefore there must have been things that were missing. And he says Darwin's theory rests partly upon the assumption that in the succession of ages, just those transition types have dropped out from the geological record, which would have proved the Darwinian conclusions had these types been preserved. Okay, now this is the most important point, which is that when it comes to the fossils that are missing for Darwin's theory to be correct, it's not just that they're missing, it's that they are selectively missing. First of all, let me just mention this point about with the contemporary paleontologist Michael Foote, who's an American paleontologist. So he has confirmed that what we have, yes, of course, in terms of every single animal that has lived on this earth, we don't. There's is a very, very small proportion that have been fossilized. But in terms of every type of animal that has existed on this earth, we have samples of every type. And we have a good representative sample, and that's all you need. And what's the proof for that? The proof for that is every time we discover new fossils, they always fall under existing types. It's not like every time we discover a new fossil, it's like, oh, this is something new, and now we've got to fit it into a different picture, a different pattern. No, it always falls under one of the existing types that we already knew about. So that proves that our knowledge of the pattern of development of life is complete. So he says in a 1997 mainstream science article, we have a representative sample, and he said, and therefore we can rely on patterns documented in the fossil record. And the reality is, with the second point as well, which is the second feature of fossils that disagrees with universal common descent, and that's stasis, is that when animals appear abruptly in the fossil record, we see examples of descent, right? So this is, we see what we would assume to be descent because it's the very same animal, right? So you see the same animal, and then within rocks that are higher up, you see the same animal again, and then within rocks that are higher up, a bit more higher up, you see the same animal again, and they stay the same, right? They stay the same, and then for extinct animals, they just suddenly disappear. So that's called stasis. They're not transforming, they're not changing. So that shows, at least to some level, that just because things are descending from parent species, they don't necessarily undergo transformations. They actually stay the same. And you have a whole set of examples called living fossils. Fossils that go back, they all the way back to the Cambrian 500 million years ago, which are exactly the same as they are then today. They've undergone no transformation. Right, so this is what David M. Rope, a very eminent 20th century paleontologist, instead of finding the gradual unfolding of life, what geologists of Darwin's time and geologists of the present day actually find is a highly uneven or jerky record. That is, species appear in the sequence very suddenly, show little or no change during the existence in the record, and then abruptly go out of the record. All right, so Agassi's point was that what you have is a type of animal which is fully formed and has all of its features all of its specialized adaptations, all there. And then you have a different type, which Darwin assumes descended from the previous type. And that's also fully formed. That also has all of its specialized features. And we've got good samples of both of these types, right? And we see many different fossil representations of them, yet we have nothing in between. The first type leading to the other type. We don't have any fossils, what was called transitions, going from the first type to the second type. Why is it that it's selectively missing? So first of all, the argument that it's incomplete is not accepted because it's complete. We have we have a good representative sample. Obviously not of every single animal that has ever existed, but every single type of animal that has existed, we have a good representative sample. And secondly, even if there were transitions, why is it only those transitions that are missing, which would prove the Darwinian theory? That's Lewis Agassiz argument, and it holds true today as it did then. And it's a very powerful argument, very logical argument. Right, the third feature of fossils that goes against universal or goes against the evolutionary story is the top-down appearance. Right? To understand this, we need to understand a little bit about classification, the field of the taxonomy, how animals are classified. So classification, just what it means is that there are some animals that are more similar or have certain features that are more similar to each other than other animals. So for example, if you take a human being, a bird, and an insect, so you wouldn't group, you'd group all of them separately if you're looking at mammals versus other things. So human beings are mammals and birds are not mammals. Whereas if you're grouping them together in terms of vertebrates, with those that have backbones and those that don't, then you'd group human beings and birds together. And you'd group insects separately because they're invertebrates. But then if you're grouping them all according to which ones are animals and which ones are not, then you'd group all three of them together. You'd say human beings, birds, and insects are all animals. Right, so there's different layers of classification or different levels of classification. The highest level of classification is called a phylum, and the plural is phyla. All right, so a phylum is the basic architecture or the basic design of the animal, the body plan. Right, so for example, you have chordates, which is a phylum. Chordates are animals that have backbones, that have vertebrates. So under vertebrates falls birds, mammals, and so on, reptiles, fish. And then you have another phylum called arthropods. So arthropods are those that have a exoskeleton, a skeleton on the outside, like insects, arachnids, spiders, crustaceans, and so on. So that's another phylum. And then you have many different phyla. And by the way, you have representatives of the majority of the animal phyla all in the Cambrian. So that's the highest level of classification, the phylum. And after the phylum are the classes. You have a class. So for example, the phylum of chordates or vertebrates have different classes. So birds are one class of vertebrates, mammals are another class, reptiles are another class, fish are another class of vertebrates. And after classes, you have orders. Orders like animals, mammals can be divided into different orders, like carnivores, rodents, primates, and so on. And then those orders can be divided into families. So carnivores, one family within carnivores is, for example, the cat family. And then those families can be divided into genera, or singular is genus. So the cat family, for example, you have the big cats. One of them are called the Panthera. And the Panthera genus can then be divided into species. For example, you have lions and tigers, which are species within the Panthera genus. Right? So this is a system of classification. And by the way, the first one, the one who came up with the modern system of classification, and then he was only developed after him, is Carl Linnaeus from, I believe, from the late 18th, early 19th century, if I'm not mistaken. And he was a creationist. He believed in the independent creation of each type of living thing. So, some evolutionists will say, oh, look, classification itself, taxonomy itself proves evolution. See, a lot of the things that are studied in science, in biology, are interpreted in light of evolution because that's the reigning paradigm, but they don't really prove evolution. It's just they're interpreted in that way. So that the first taxonomist was not an evolutionist. So what evolutionists believe is that what you have is the higher orders of classification appearing later because of an accumulation of changes from or within lower orders of classification. So Richard Dawkins, he presents a typical evolutionary point of view. What had been distinct species within one genus became, in the fullness of time, distinct genre within one family. Later families will be found to have diverged to the point where taxonomists prefer to call them orders, then classes, then phyla. And the reality is the complete opposite. So that's the theory, and you can understand why that's the theory, because remember, they believe in things happening by accident, by natural processes. So you'd expect there to be simplicity, and that simplicity becomes more and more complex as time goes by, because this is the reductionist, materialist point of way of approaching things. So Richard Dawkins is not a paleontologist. So you have real paleontologists who are experts in fossils, Douglas Irwin, James Valentine, and they write, this is 10 years before Richard Dawkins actually wrote this, so this was already known. He said that they said the fossil record suggests that the major pulse of diversification of phyla occurs before that of classes, classes before that of orders, orders before that of families. The higher taxa do not seem to have diverged through an accumulation of lower taxa. Meaning the higher systems of classification do not seem to have emerged from the lower levels of classification. Right. So as I mentioned, the Cambrian, which is when complex animals originate, you have representatives of pretty much all of the phyla in the Cambrian, and only then you have classes and orders and classes and families belonging to those phyla emerging later on. And this is important because what it shows is a top-down pattern. And a top-down pattern is exactly how you'd expect an architect or a designer or an engineer to make something. So you have a model or a basic design or architecture of something, and then you make you have things that fall within that basic design, or other models that will fall within that basic design, like subtypes. Right, so this is one example of where the facts oppose the theory. So just to very quickly come back to that question, so if they are from the same ground, then would we say that they are descended? No, we wouldn't, because, as I said, because of these facts, right? Because when one type of living thing appears, like for example, when a turtle appears, even if it's in the same ground as other animals, you don't see anything leading up to a turtle. And when the turtle appears, it appeared 200 million years ago, according to their dating techniques, it stayed the same throughout. It hasn't changed in that period. Now, what the evolutionists would point to is they'll say, okay, maybe that is the general pattern, but there are examples that we can point to where we see one type of animal changing into another type. And they will point to certain examples, but all of these examples suffer from problems. And all of them are their own interpretations. And interpretation based on is where the theory comes before the facts. So they present the fossil evidence in light of the theory rather than what they actually show. And one example of this, I did want to go into this in a bit more detail, but I think we'll just go through it quickly because there's other things to cover. The example of whale evolution, or the fossils of whales. So in the 1990s, there were all of these discoveries of different fossils that they found within rocks that are dated close to when the first fossil whale appears. All right, so the first fossil whale appears, and it's called Basilosaurus. Right, so this is a fossil whale. And in the 1990s, they found other fossils that around that same time. These are up in the Pachycetus, Ambulocetus natans, Myocetus, Cuchicetus, and Rhodiocetus. Okay, cetus just means whale. And these are names. All right, so Ambulocetus natans means walking, swimming whales. Pachycetus means Pakistani whale because they found a fossil in Pakistan. Right, and the reality is none of these are actually whales. Basilosaurus is a whale. Pachycetus is more like a wolf, and it lived fully on land. And the other examples, most of them found in the 1990s, are of amphibious mammals, like otters or sea lions that live partly in the water and partly on land. All right, so they live mainly on land. So they're really land mammals. And whereas whales are fully aquatic mammals, they live fully in the water. They can't live outside of the water. They would not survive outside of the water. And there's a lot of changes and transformations that need to take place in order for a mammal that is a land mammal to become a fully aquatic mammal. So in reality, these are all independent types of animals, but it's just interpreted in a way to make it seem that they are transitions because they appear in the times that they would want them to. And so Stephen Jay Gould, who's a very famous paleontologist from America, he's pretty much, you could say, the American Richard Dawkins in terms of trying to communicate Darwin's ideas in popular books and so on. So he said about this whale transition is the sweetest series of transition fossils an evolutionist could ever hope to find. Right. But as I said, the reality is what I mentioned. These are not whales, and they don't look like anything that is leading up to a whale. And apart from that, just a couple of years ago, they found a fossil, I think around Argentina, of a whale. There's a full whale that is dated before these supposed intermediaries, before Ambulocetus natans, before Myocetus, before Cuchicetus, and so on. So the whales actually existed before them, or at the same time as them. So they could not have been transitions. So that this is an example of where they present something as a clear, a series of transitions from one type to another. But when you look at it a bit more deeply, you scrutinize it a little bit more than at the surface level, you find that it's completely without any substance. Now, sometimes they point to human fossils or fossils along the human line, and they say, oh, look, clearly we got fossils that are leading up to modern humans. Now, there's a whole field devoted to the fossils that are in the human line. So these are called hominin fossils. Hominin just means fossils that are of. See, they believe that human beings and chimps share a common ancestor. So from that supposed common ancestor, when the human line is supposed to have diverged, become separate from the common ancestor between humans and chimps, the fossils that fall within that line are called hominin fossils. So those that specialize in that are called paleoanthropologists, meaning paleontologists who specialize in this category of fossils. Now, what they believe is that human beings are related, or are most closely related to the great apes, and specifically the chimpanzees. So the great apes are, chimpanzees, gorillas, orangutans, bonobos. Right? So these are the great apes, and they're different from monkeys because monkeys are they're also primates, but monkeys are smaller and they have tails, whereas these great apes don't. So they say that from all of these great apes, the human beings are most similar to the chimpanzees. So that's the theory. Now, what's the fossil evidence? Now, the fossil record and real experts, real specialists in this field will say, as opposed to popularizers, as opposed to people who distort the evidence and misrepresent, they will say, real experts will say that the fossil evidence is very, very incomplete and very fragmented. We don't have much evidence. But even from what we have, take an objective look at it, it paints a very clear picture. And the very clear picture is that when the human species occurs, and they call it Homo, when the Homo species arrives, it arrives suddenly without any connection with the previous species. So you have these group of fossils which are called Australopithecines. Right? Australopithecines, I believe it just means southern apes. Right? And then you have Homo, and Homo just means human. So yeah, you have these different types of Homo. You have Homo erectus, Homo neanderthalensis, like the Neanderthals, and then you have the Homo sapiens. And Homo is basically human. They look completely human. And Australopithecines are pretty much apes, right? So this is Leslie Aiello, who is a Italian-American paleoanthropologist. So he said that Australopithecines are like apes, and the Homo group are like humans. Something major occurred when Homo evolved, and it wasn't just in the brain. So one of the things that's significant about the Homo fossils is that the skull shows that the brain was a lot bigger. So with humans, the brain is a lot bigger than with apes. With Australopithecines, some of you probably heard of Lucy, right? So Lucy was an Australopithecine, and you have these drawings, these caricatures, and those drawings are embellishments. So obviously, the fossils are just bones, right? And the features, the facial features and emotions and so on, these are just embellishments from the person who's drawing. So often Lucy is painted walking on two legs, and often with watery eyes, like as if she's crying. Remember, chimps can't cry. as in they can cry out, but they can't shed tears. Shedding tears is unique to human beings. So they would present Lucy in this way. Whereas Lucy was just an ape. She based on the evidence, she's Lucy is Australopithecine. It would climb on trees. It would walk on. It was a knuckle walker. It walk on its knuckles. Walk on all fours. It was much smaller. It's smaller than humans, and it's completely ape-like. And it doesn't have the distinctive ear bones that human beings have, as opposed to Homo erectus, Neanderthals, and Homo sapiens. Now, the only major exception to this is something called a Homo habilis. So Homo habilis is something that they used to believe is transitional between Australopithecines and Homo erectus. Homo erectus is the earliest Homo. And but now they find that Homo habilis actually was coterminous, meaning was at the same time as Homo erectus. So it couldn't have been a transitional. And plus, Homo habilis, based on recent studies, appears to have just been Australopithecines. They were just apes. All right. So 2015 science paper states postcranial remains of Homo habilis appear to reflect an Australopith-like body plan. John Hawkes, a paleoanthropologist from America, he says, no gradual series of changes in earlier Australopithecine populations clearly leads to the new species, meaning Homo. No Australopithecine species is obviously ancestral. This may seem unexpected because for 3 decades, habiline species, meaning Homo habilis, have been interpreted as being just such transitional taxa, linking Australopithecus through the habilines to later Homo species. But with a few exceptions, the known habiline specimens are now recognized to be less than 2 million years old, and therefore are too recent to be transitional forms, leading to Homo sapiens. Here's another paper. Whatever its time and place of origin and direction of speed, this species, meaning Homo erectus, dispersed widely and possibly abruptly before 1.5 million years. This is according to their dating techniques. 2016 paper, although the transition from Australopithecus to Homo is usually thought of as a momentous transformation, the fossil record bearing on the origin and earliest evolution of Homo is virtually undocumented, meaning it doesn't exist. All right, so there's nothing between Australopithecus and Homo. It's just assumed that it does exist. A 2000 paper, there is a period of very rapid evolution corresponding to the emergence of the genus Homo. Ernst Mayr, very, very famous evolutionist from the 20th century, he wrote in 2004, the earliest fossils of Homo, meaning Homo erectus, are separated from Australopithecus by a large unbridged gap. Not having any fossils that can serve as missing links, we have to fall back on the time-honored method of historical science, the construction of a historical narrative. Meaning we just have to make things up. A 1999 paper, skeletons of Homo neanderthalensis, meaning Neanderthals, indicate that their body shape was within the range of variation seen in modern humans. And Neanderthals and modern humans are members of the same species who interbred freely. Okay, and this is another thing with Neanderthals. Normally, the way they are represented is more ape-like. They present them as, you know, like bumbling fools, beating their chest and so on. But really, they were just human beings. They were just normal people with just a bit more, with protruding bones from the eyebrow bones, and probably a bit stockier. But they actually had as this quote mentions, their skeletons fall within the variation that we see within human beings today. And same with Homo erectus. So Homo erectus and so Homo, they're the first ones that walk upright, right? They walk on 2 legs. And human beings appear to be designed to walk and run. And apes are designed to climb trees and walk on all 4s. Now, this moves us on nicely to this issue of human-chimps dissimilarities or similarities. See, what the evolutionists try to push and portray is that humans and chimps are very, very similar to each other. And the reality is that they are very, very different in their anatomy, in their physiology, in their behavior, and so on. Just to give a quick list, the teeth of human beings develop a lot slower than in chimps. Human beings have a lot less hair than chimps. Human babies take a lot longer to mature than chimps. So human babies, they start out very, very helpless. Those of you who are parents, I'm sure would be very familiar with this. Babies are very, very helpless. And this is quite different from chimps. So chimps, very, very quickly, they learn to take care of, become independent and learn to take care of themselves. Human babies are very helpless. And only after some time become independent. And Allah refers to this in the Quran. Allahu alladhi khalaqakum min dha'fin, thumma ja'ala min ba'di dha'fin quwwatan, thumma ja'ala min ba'di quwwatin dha'fan wa shayba. Allah created you and made you start out being very, very weak and helpless, and then He gave you this strength. After strength, He gave you weakness again. And the human brains are much bigger. Human beings can get AIDS and malaria. Chimps can't. Humans shed tears. Chimps don't. Humans can swim. Chimps can't. Humans have chins. Chimps don't. Humans walk and run upright. Our feet are different and designed for walking, while chimps' feet are designed for climbing. We have longer necks. Human hands are designed for tool use, while chimps are designed for knuckle walking. Humans have a poorer sense of smell, but a greater sense of touch. And of course, in terms of intelligence, human beings plan and think about the past and about the future. Human beings are uniquely capable of language meaning with grammar. And we have this, we have this ability of delaying gratification. Right? So and Islamically that's called sabr. Sabr, one aspect of sabr is self-control. Right? That you see something that you want immediately, that you're going to get some short-term benefit from, but you realize that in the long term, this is going to be harmful for me. So you exercise control and sabr. This is unique to human beings. Chimps can't do that. If they see something that they want, unless there's an immediate danger that's stopping them from that, they're going to go and get it. They can't delay gratification. Okay. Now, what they will point to is this similarity, what they say that humans and chimps have this 1% similarity in the genomes. Now, to understand this, you need to know what genome is and what genes are, what DNA is. Okay, now this might get a little bit technical, but I'll try to keep it simple. Now, every living thing is made up of cells. All right, cells which have a wall, and that wall makes what's inside the cell separate from what's outside of it. Now, most of the living things that we are familiar with are multicellular. They have many, many cells. But there are some animals, there's even some. All right, so there's some living things which are unicellular, right? They have single cells. Now, within the cell, living things are made up of stuff, right? You're made up of material. You're made up of things. But the things that a human being is made up of is different from the things that human beings make. Like the things that we make are made up of, like, say, wood and bricks and cement and metals and so on. Human beings are made up of stuff as well. What are we made up of? Mainly proteins. Right? There's proteins, carbohydrates, lipids. It's what's called organic matter. and proteins. So what makes our skin is proteins and hair, like keratin and collagen. So the structural material that makes up these stuff is protein, and even in bones, right? Collagen in bones is proteins. Now, what you have in all cells is DNA. Now, DNA is a molecule, meaning it's a chemical molecule, and it stands for deoxyribonucleic acid. And the basic structure of it is that it's a polymer, meaning it's made up of many different subunits. And each subunit differs. There's 4 different types of subunits. It's called 4 different types of bases. Right? And these 4 different types have names. The bases have names, and they are assigned, or they have abbreviations, and these abbreviations are A, C, T, and G. Now the DNA, these subunits can link to each other, but it's not the A, C, T, and G that links to each other in DNA, but it's the other parts of it. See that the DNA is made up of 3 parts: a sugar part, a phosphate part, and then the bases. And the bases are A, C, T, or G. So it's the sugar and the phosphate part that makes a chain that links the subunits together to make a DNA chain, or what's called a strand. And then the bases stick out. The A sticks out, the C sticks out, the T sticks out, and the G sticks out. Now, there's no determinacy, there's no chemical rule or law in which sequence the subunits will link to each other. So it's not like it has to always be ACTG, ACTG, ACTG. No, it can be anything. It can be AAA, TC, TTT, TC. It can be anything, right? So this is where you get the DNA code or the information, which we'll come to it shortly. And what the DNA strand, it doesn't exist as a single strand. Rather, each strand has a complementary strand to which it binds. All right, so bases bind to each other. All right, so A binds to T and C binds to G. All right, so if on one strand you have an A, then the complementary strand next to it you'll have a T, and if on one strand you have a C, then the complementary strand you have a G. Now the DNA molecule, you've probably heard of the double helix. That's the structure of it, and it's basically 2 strands that are winding around each other like a winding ladder. So that's the structure of DNA. Now, I think there's 2 remarkable features of DNA. One is DNA replication, and the other is protein synthesis. Meaning, protein synthesis just means making proteins. Okay, now DNA replication, what that is that DNA can make copies of itself. So as I mentioned, it exists as 2 complementary strands. So what happens is the 2 strands are separated from each other, as in they're unzipped, and then you get nucleotide bases, meaning DNA subunits attaching to each of the 2 complementary strands, and then instead of having 1 pair, you end up with 2 pairs. So as in DNA replicates itself. All right, so that's DNA replication. But this process of DNA replication, it doesn't happen by itself. It's very, very tightly controlled, and it cannot happen by itself. As in, DNA can't do it on its own. There has to be a system, a huge mechanical system in place that makes it happen. That first of all unzips and these are actually done by proteins. So proteins are also function as enzymes. These are like nanobots, machines at a very, very small level that are pulling these chemicals, these strands apart, and then allowing the creation of another set of DNA molecules. That's DNA replication. And then protein synthesis, the way that happens is the DNA strands opens up a bit, and then you have this other molecule called RNA. RNA is very similar to DNA, but instead of it has other differences as well. But one of the differences is instead of a T, you have a U. So wherever you have an A within the DNA molecule, a U from the RNA will attach to it. And wherever you have a C, a G from the RNA will attach to it. Wherever you have a T, an A from the RNA will attach to it. And wherever you have a G, a C from the RNA will attach to it. And then you can make an RNA molecule that is pretty much a copy of a DNA, of a strand, of a section of DNA. And that's called mRNA, the messenger RNA. Now, this messenger RNA, once it's transcribed or once it's copied a section of the DNA, it will be transported to a machine called a ribosome. Okay, so now the ribosome is like a clamp. It holds the mRNA in place, and then you have another molecule called tRNA, which is RNA, but it has a different structure. So this RNA, tRNA, what it does is it has an amino acid. Okay, first of all, proteins are also molecules. And they're made up of amino acids. So just like DNA is made up of subunits, A, C, T, and G, proteins are also made up of subunits called amino acids. And these amino acids, there's about 20 of them, a bit more than 20 of them. And they have different features. Okay. And the thing with proteins is it's all to do with the arrangement of the amino acids. Okay. So when the amino acids and a typical length of a protein sequence would be, say, about 300 amino acids. A very small one would be about 150. But typical length is about 300 amino acids. And then when you have that, then what happens is the amino acids, if they have the correct sequence, then they will link to each other. Yeah, you have different types of bonds, right? You have chemical bonds, you have electric bonds, and other types of bonds which will make the sequence of amino acids fold into a certain shape, right? And what's important is that, or something that is important, is that majority of amino acid sequences, if you just put together a random sequence of 150 amino acids, the majority of them, 99.99999% of them, will not fold into a functional sequence. It won't do anything. It's just useless junk. So the folding has to be done as in the sequence has to be very, very precise so that it can fold into a precise functional tool. It can become a tool. So all of these things are tools with very specific functions. Again, all of this is design language. Now, so what happens is with the mRNA, remember mRNA has just basically copied the DNA, what's in the DNA. The tRNA, you have different tRNAs, and each one, so you have just about 20 of them. Each one of them connects to one type of amino acid. And then they, according to the code that is according to what's on the mRNA, a tRNA will attach to it. All right, so the way it works is that every three letters of the mRNA determines one amino acid. right? So, so for example, one amino acid is glycine. That's the name of an amino acid, glycine. And if you have GGU on the mRNA, what was complementary to that is CCA. So the tRNA will have a CCA at its bottom and a glycine at the other end. So the CCA will attach to the mRNA at that point, and then wherever you have the next triplet code next to it, and whatever that is, the tRNA that has the complementary amino acid will attach to that, and like that, the amino acids will, glycine, alanine, all of them, whatever they are, will all line up in a very, very fixed sequence. All right, so this is a set of instructions in the DNA that is coding for. So the DNA is basically like a language. Right? It's just code. It's just information. There's nothing structural to it. Well, there is a structured element to it, as in it has to be compacted and put into a very compact, in a very compact way. But it doesn't really do. It doesn't have any functions like proteins do. But it's a code. It codes for a specific sequence of amino acids so that they can fold into a functional sequence. Now, of course, this is something remarkable. And every living thing has DNA. And so that shows to us that information is built in to the very fabric of life. And this problem of information, obviously evolutionists, materialists, reductionists, they just generally ignore this problem, even though it's so obvious. But there's many other layers of problems that face the materialist. One, for example, is, how does this whole system arise? DNA replicates, yes, but DNA doesn't replicate by itself. It needs proteins. And how do you get proteins? You get them through the instructions in the DNA. Right? So you don't have proteins without DNA, nor can you have replication of DNA without proteins. So they can't exist without each other. So they have to all come together at once. So this is, and that's literally impossible in terms of natural processes. Right, now we can sort of understand this. So a gene is basically a stretch of DNA that codes for a protein, or codes for one protein or more. And a genome is the full genetic instructions within one organism. So you might have heard of the Human Genome Project, which is sequencing the human genome. So in terms of A's, C's, T's, and G's, it's just a long set of letters. And they have also sequenced the chimp genome. So now what they do is they've compared them, and some of them have said, "Oh, look, there's 99% similarity." Now, what they mean by that is they get sections of the genome which look similar between humans and chimps, and then they align them, and they find that those sections that they have aligned are very, very similar to each other, and there's changes in certain places which amount to, say, 1% change. So really what it amounts to, this 99% similarity, is similarity in certain sections of the genome which we wouldn't contest, because there are similarities. We see similarities in the morphological and anatomical level as well, not just in the genome level. But we would contest this 1.1% or 1.5% figure. And scientists, mainstream scientists, have done so as well. One science paper, and this is, I believe, in Nature or Science, one of the famous science papers, in 2007, they had an article by the name of "The Myth of 1%." And another paper in 2006, right? So see, what you have is in the human genome, there are certain genes that are represented multiple times. So the same gene, so you might have a gene in a chimpanzee that only appears once, but in the human being appears more than once, appears several times. And sometimes you have a gene in the chimp that is present in the chimp, but is not present in the human being. This is not accounted for in that 1% figure. So this research in 2006, what they said based on these facts, said our results imply that humans and chimpanzees differ by at least 6% in their complement of genes, which stands in stark contrast to the oft-cited 1.5%. All right, so but even 6%, you might say, okay, 6% sounds like a lot, or sounds like very little. You know, does that mean we're 94% similar to chimps? The answer is no, but this is because of a misunderstanding, which is sometimes called, you know, genetic determinism or genetic reductionism, as in you reduce the living thing to its genome. You can't reduce a living thing to its genome. A living thing is far, far, far more complex than just its DNA. Of course, the DNA is one example of a degree of complexity, but the DNA doesn't explain everything. The DNA is only part of the explanation of the complexity in life. All right, so one example is this thing about one gene, right? So just because you have a gene that's, okay, they might even be identical, it doesn't mean that that gene will code for the same protein. Right? There's something called splicing and alternative splicing. So which is, what happens is that the mRNA, so the RNA that it transcribes the code in DNA, once the mRNA is created, parts of it are cut out. That's called splicing. Parts of it are cut out. And then those parts of it that are cut out will join onto each other. But they can join onto each other in different ways. So that's called alternative splicing. And because of the different way that they are joined on, then you can get different proteins that result from translating those mRNAs into proteins. All right? And so because of this, through alternative splicing, even from just one gene, you can get thousands of proteins, thousands of different proteins. And this has actually been proven in certain animals that they have studied. And a 2007 paper describes an important role for alternative splicing in establishing differences between humans and chimpanzees. So Anne Geiger, okay, so this is not from a mainstream science paper. Anne Geiger is not a mainstream scientist. It's important to say that because she is a scientist who is fully well credentialed. But the thing is, anybody who challenges the evolutionary worldview in biology, they are sidelined. And especially if they adopt intelligent design, Anne Geiger is a supporter of intelligent design who believes that the only way you can explain biological complexity is by the activity of an intelligent designer. So she wrote, one can think of our genes as being like the bricks and mortar, nails and wood, shingles and wires out of which houses are made. Two houses may look different, but be composed of the same basic building blocks. Right? So you can't reduce a living thing to its DNA, to its genome. So even if we say that there's only 6% differences, and in reality there's probably more between the chimp DNA and the human DNA, but there's layers upon layers of complexity and code beyond just the DNA, and these are just being discovered, just recently. Like there's something now which they call the sugar code, which is not the DNA code, it's the sugar code. So what happens is when a protein is made, then there are sugar carbohydrate molecules which attach to the protein. And you also have sugar molecules which attach to the surface of the cell. And the surface of the cell membrane, is made up of lipids. And they say that this arrangement of the sugar molecules and the sugar molecules themselves are another level of code, which tells the cell how to behave, tells the proteins where to go, and so on. Now, one thing that before I mention this, actually, something important is that in the 19th century, see, this is something completely unknown to 19th century biology, the complexity of the cell. So what I've just described to you is one level of the complexity of the cell, right? About the DNA and the proteins and so on. But the cell truly is a marvel of complexity. See, in the 19th century, they used to believe that the cell is simple. It's just a blob of jelly. And some of them used to even believe that a cell or life could just spontaneously arise, just generate, just come out of nowhere. But really, the cell is extremely, extremely complex. It has portals, it has highways, roads, it has cargo that's shifted, it has postcodes, it has addresses, and so on. And it has machines. And some of them, and now they can actually image them. They've got these imaging techniques by which you can image at that level. Obviously, with the microscope you can't, but then there's other types of imaging with which you can. So you see, they used to believe in the early 20th century, because they're materialists and reductionists, they used to believe that once we get to the base of life, which they used to think is proteins, that it becomes simple. All right, so proteins are probably just little square boxes, and then the complexity of life emerges at higher levels. So when the first protein was actually viewed, by this imaging technique, I think is in the 1950s, and the first person who showed an image of the first protein that was imaged in this way, I think it was myoglobin. He won the Nobel Prize for it. When he saw it, he said, I was horrified. This looks horrendous. It looks hideous. Because when you look at an actual protein, the idea behind it is just like the mechanics. Like if somebody looks at a car, a child who looks at a car might think, oh, maybe just like on the outside it looks, you can put it into some conceptual framework. Maybe on the inside it looks that simple as well. But when he looks at the engine, it looks very, very complicated. In the same way, a protein is very, 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 very complicated. And a picture of it, like an enzyme, like a myoglobin. Myoglobin is not an enzyme, but it looks like, say, a ball of strings just tangled up in a very, very specific arrangement, but without symmetry, without any fixed arrangement that would always be there. It's very, very, as in, it's not it's heterogeneous, it's asymmetrical. And myoglobin was actually a simple protein. And now you can actually look at other types and see proteins don't just work individually, they work in groups. A majority of them. And many of them literally look like machines. So one of the most famous ones is the bacterial flagellum, which you can check up, the bacterial flagellum. And you can even see an electron micrograph, an image of it at that level. And it looks like a machine. It looks like a nano robot, a nano machine, a machine at a very, very small level. And it actually is a machine. It's a rotary device. It's a motor that's connected to a propeller, like a whip-like thing at the back of the bacterial cell, and it rotates that propeller, and by means of that, the bacterium can swim. And all of that is assembled by instructions within the cell itself. And you can imagine, because it's not haphazard, it's not random, it's very, very non-random, it's very, very precise. Because if you had one protein part coming out before another, whereas it's supposed to be in the reverse order, then the system, the machine wouldn't work. And even those individual parts are very, very complex. Now, see, they would make a family tree. Phylogeny just means a family tree, a history of how living things are related to each other. So they would say, for example, humans and chimpanzees are more closely related to each other than are humans and gorillas. So that's an example of a phylogeny. So normally the way that they would do that is based on characters that you see morphologically. So you would take the eye, for example, and say, okay, based on which animals have this type of eye, we will say that they can be branched together, and they're more closely related than those that are otherwise. So taking certain parts and comparing them to which ones have more, which are more similar in them to which are not, you can create phylogenies. And what they thought is that the phylogenies that are made based on morphology, based on the anatomy of an animal, will agree with the phylogenies that they make using the molecules. So molecules, so for example, proteins, they have a sequence, right? So let's just take myoglobin as an example, because we mentioned that. So myoglobin is a protein that has a certain sequence. Now, other animals also have myoglobin, but their sequences differ slightly. So based on how they differ, which ones are more close to others, you can construct a family tree and say this one is more closely related than others. Or you can even use DNA, you can use proteins, you can use ribosome, which is RNA, or partly RNA. But what happens is you get different phylogenies depending on which molecule you use. And this completely shatters the whole picture of the tree of life and universal common descent. All right, so here, there's a quote here, and this is the norm. This is typical. So phylogenetic conflict is common and frequently the norm rather than the exception. Incongruence between phylogenies derived from morphological versus molecular analyses and between trees based on different subsets of molecular sequences has become pervasive as data sets have expanded rapidly in both characters and species. Now, an argument that is presented by people who support this universal common descent is that we have things that are useless within living things. And why would we have things that are useless if they were designed? All right. And sometimes they say that the reason why we have it is because these were just derived from existing types of living things which did have a use for them, but now it's just a historical remnant that has remained but without any use. And one famous example that's mentioned is the appendix, which is called it's referred to as a vestigial organ, meaning something useless that's something left over from our evolutionary history. But now it's found that the appendix is not useless. All right, so this is a 2004 paper, long thought to be an evolutionary remnant of little significance to normal physiology. The appendix has more recently been identified as an important component of mammalian immune function. And another example is junk DNA. You see what I mentioned about DNA, that it codes for protein. That's only about 2% of the genome that does that. The 98% of the genome does not code for protein. So they used to think that the majority of the genome is useless, is junk, as in it doesn't do anything. But now they found out that it does do things. right? So Francis Collins, who's an evolutionist, and he was the leader of the Human Genome Project. He says, well, he used to use junk DNA as a proof of evolution, but now now he's changed his tune, which is to his credit, because he's accepting that evidence that I used is no longer valid. So he's got this famous book called The Language of God, where he tries to push this theory of evolution. And this is one of the arguments that he used. But now he's taking it back. He says, in terms of junk DNA, we don't use that term anymore, because I think it was pretty much a case of hubris meaning like arrogance, to imagine that we could dispense with any part of the genome as if we knew enough to say it wasn't functional. So remember, this is an argument from ignorance. To say that this thing is not useful is an argument from ignorance. Most of the genome that we used to think was there for spacer turns out to be doing stuff. So most of the DNA, even those that don't code for proteins, most of the DNA is actually transcribed, meaning it's made into RNA. And the RNA that is produced from the DNA is doing stuff. It actually does things. It has many, many different roles. And I won't go into some of them. I've made a note of some of them, but I won't go into it now. Right. Another thing that they point to is what it looks like is it doesn't look like it was designed. And the famous example is the vertebra eye. I think we are running out of time, so I won't actually go into this. Right, so that's universal common descent, which suffers from many, many, many problems. And in fact, a face value reading of the evidence would clearly indicate to us that each type of living thing was created separately with its own specialized features and adaptations, independent of other types of animals in terms of being related to them with ancestry or descent. Now, in terms of the second part, of evolution, which is that transformations and new characteristics and novelties can occur by natural processes. The main process that's normally invoked is natural selection and random mutations. Now, the first thing about natural selection is natural selection doesn't do anything. It doesn't make anything. All right, so just look at the logic of it. I'm sure all of you know what natural selection is. It's also called survival of the fittest. Those animals that are more likely to survive or more fit to survive will survive. So first of all, that's a tautology. Those that will survive will survive. It's not really saying anything. And secondly, those that will survive doesn't create anything. So natural selection doesn't, as I wrote here, doesn't construct but conserves. So things that are not fit enough to survive will just die out. And those that are fit enough to survive will live on, will survive, will reproduce. That's natural selection. It doesn't do anything. So where does the construction part of it come from? All right, so some have said, natural selection might explain the survival of the fittest, but it doesn't explain the arrival of the fittest. All right, so the way they explain that is by mutations. And mutations are, so as I said, you see the DNA code is very, very specific, and it will code for proteins. And sometimes when the DNA copies, there's mistakes in copying. So a DNA doesn't copy itself exactly. There's an accident that happens. And this is called a mutation. And sometimes a mutation can result in a different protein or a different sequence to the protein. And these will cause changes at the bigger level. So the bigger level is called the phenotype. The genotype is at the sequence level, and the bigger type is, or the morphological and anatomical and the gross level is called phenotype. So mutations do create things, but mutations are don't construct anything. There's no demonstration, no observation of mutations really doing anything constructive. Okay, and they will point to some examples. Okay, so one example is the four-winged fruit fly. Right, so fruit fly already has wings. Right, it's got wings, but what happened was because of this mutation, there was a misfiring, and wings were made where it was not supposed to have been made. So those wings didn't have any muscles to be able to fly them, and they were just dead weight. So that animal in the wild would not survive. And the important thing is with this mutation, the four-winged fruit fly, they weren't actually creating wings. The wings were already there. The assembly instructions, the instructions for making wings were already inside the fly. It's just that they sent that instruction by a mutation to the wrong place. If they, by means of mutation, created wings in a pig or in a whale or in a lion, then we'll say, okay, that's amazing. You've done something constructive there. But here you're creating wings in an animal that's already got wings, just in the wrong place, and it's useless, and you would die in the wild. And they've been mutating fruit flies for ages, and all they get really is either dead fruit flies, normal fruit flies, or defective fruit flies. Another example is bacterial resistance. Right, there's different types of bacterial resistance. So it means resistance to antibiotics, and antibiotics work in different ways. So one antibiotic is penicillin. So penicillin, in some bacteria, they already have an enzyme called beta-lactamase, and beta-lactamase inactivates the penicillin. So those bacteria that already have this protein, beta-lactamase, they will survive, and those that don't have the protein will die out. So then what ends up happening is that the bacteria that do have that protein will become dominant. So that all that's happening there is just one type of bacteria is becoming is now proportionally bigger in terms of population size to the other types of bacteria. And sometimes what happens is, see, there's one type of antibiotic called streptomycin. And streptomycin, the way it works is it binds to the ribosome. Remember, the ribosome is the clamp that holds the mRNA in place in order for tRNA to attach to it and create the protein, the amino acid sequence. So that ribosome, the streptomycin attaches to that and deactivates it. So what happens is a mutation happens in some bacteria which breaks the ribosome a little bit. It just damages a little bit. So and because of that little damage, the streptomycin can't bind to it, and therefore it survives. It doesn't damage it so much that the ribosome is completely malfunctioning, but it just stops working. I mean, it's just a slight damage, but that slight damage is advantageous in the condition of antibiotics. So as you can see, it's breaking things down. It's not creating anything, and this is the norm. 2014 science paper for microbes. Devolution is evolution, as in breaking things down. Deconstruction is how evolution works in what we have observed in bacteria. All right, I was going to go into dogs. Now, there's different dog breeds. Again, these are all examples of breaking things down at a genetic level, at an anatomical level. You have different races of dogs. You have some that are really dwarves, and some that suffer from giantism. They become excessively big, and that's because the growth hormone, which is supposed to stop, doesn't stop. So it's actually breaking something. It's not creating anything, and that causes problems. So it's not true that they have knowledge of how novelties are created. So this is a 2006 mainstream science paper, how fundamental innovations originate in evolution remains one of the most enigmatic questions of biology. And you see the the material of life, as I said, organic molecules, DNA, proteins, lipids, carbohydrates, they are very, very complex in themselves. And they don't just, and so for example, outside of living things, you won't randomly get a protein just coming out of nowhere. You might accidentally get an amino acid, but even that is unlikely in the prebiotic atmosphere, meaning before you had life, before living things existed on Earth, it would be very, very unlikely, probably actually impossible for, in that atmosphere, for even amino acids by accident to be created, meaning the subunits of proteins. Forget about protein, and forget about a correct sequence and a correct functioning protein molecule. You won't even get amino acids. That's how impossible these origin of life scenarios are. And this is from James Tour, who's a synthetic chemist from America, who's at the top of his field. He works on making nano technology, and he even made a car, he called it a nano car. And he says that what's the molecules that you have in living things, these nano cars are nothing compared to them. So this is what he says: is designing nano cars is child's play in comparison to the complex molecular machinery and information processing systems that work in the synthesis of proteins, enzymes, DNA, RNA, and polysaccharides. Polysaccharides meaning sugars and carbohydrates, let alone their assembly into complex functional macroscopic systems. Those who think scientists understand how prebiotic, meaning before you had life, chemical mechanisms produced the first life, are wholly misinformed. He wrote this in 2017, just a few months ago. Okay. So as I said, just by arranging a sequence of amino acids together at random, the possibility that it will form into a functioning protein is literally impossible. Right? Given how rare it is, and given the amount of time that we have on the Earth, even if we said that, basically having a protein with the correct sequence is like a meaningful paragraph. It's a meaningful paragraph with meaningful sentences. So if you imagined they say the universe is 15 billion years old. For 15 billion years even, you had a monkey or two randomly banging on a keyboard for that amount of time, or even if you had 1 million or 1 billion keyboards that they were banging on for that period of time, you might get a few words here and there, and at most maybe a phrase that occurred by accident, but never will you get a paragraph. Never will you get a paragraph. In the same way, you will never get a protein by accident, just by the random association of amino acids, because that's how functionally meaningful and complex it is. It's at that level, probably even more complex than a paragraph. So Francois Jacob, who's a French scientist, a very prominent one from the 20th century, he was a materialist, but he believed that proteins are created, new proteins are created by a variation from old types. So old types undergo mutations, and that creates new types. So he says the probability that a functional protein would appear de novo by random association of amino acids is practically zero. Meaning obviously it's not aqlan, it's not conceptually and rationally zero, but adatan, meaning realistically speaking, physically speaking, it's impossible. It's a miracle. But now what they found is, now you would actually find this in scientific papers, that proteins have been created de novo. Francois Jacob, he wrote this in 1970s, but in the 2000s, now in 2010, because now what they found is, and it was called lineage specific proteins, or orphans. Orphans meaning you have proteins which do stuff, which are actual functioning proteins that are unique to a species. So octopuses have their own unique proteins. Human beings have their own unique proteins. Mice have their own, which are not found in any other species, which have no comparison in any other species. what they call no homology with proteins found in any other species, and they call them orphans. So you can imagine the level of impossibility that we're talking about. I did want to quickly mention this example because people have so much admiration and respect and look at scientists so highly. But scientists are human beings, and they have their own agendas, they have their own philosophy, their own commitments, their own beliefs. And one of these scientists is Jack Szostak, who's at the top in the field of origin of life research. And he's a materialist. So he believes that life originated by accident, by material processes. So he basically believes in the impossible. He believes in fairies and myths. But because of that belief, he came out with a result that was wrong. And he was produced in a mainstream scientific paper in just last year. Right? You see, as I mentioned, that example of DNA replication, this is a huge dilemma with origin of life studies. For DNA to replicate, you need proteins. But then to get proteins, you need DNA. Right? And it's impossible that they could have arose by accident together. So what they've come up with is an RNA world. So they say that we can just sidestep DNA and RNA, DNA and proteins, and just use RNA because RNA has some enzymatic ability. So RNA can do some of the work of proteins, and it has the same capacity as DNA to copy itself. So what they have to come up with is RNA performing or self-replicating itself, as in RNA in those two complementary strands, and then they're separated, and then you get a second pair that is a copy of the first, and then they can continuously replicate itself without outside intervention. So they produced this paper saying that, we've done this. We've shown evidence of RNA self-replicating. But then what happened was a team member tried to make those same results again, tried to replicate the results, and it didn't work. As in, it was a false result. It was mistaken. So the RNA could not self-replicate. And then he retracted the paper. He's a Nobel Prize winner, by the way. And he says, in retrospect, we were totally blinded by our belief. We were not as careful or as rigorous as we should have been. And this is not, back in the days. This is just a few months ago. So these are some books. Essential reading, Explore Evolution, by Ralph Silk, who's a biochemist, and Stephen Meyer. So this is a good introductory text, weighing up arguments for and against evolution. And then Undeniable. So this is Undeniable by Douglas Axe. So Douglas Axe is a scientist. He worked in Cambridge for about 10 years, and he's an expert on proteins. And he in this book, he presents a powerful common sense, intuition-based and scientific case against naturalistic causes of the origin of biological novelties. And here you have a list of recommended probably from all of these, to start with, I'd probably recommend Icons of Evolution and Zombie Science. Just to start, because what they present is both of them by Jonathan Wells, who's a developmental biologist and a biochemist from America. And what he does is he targets directly the popular presentations of evolution, and he shows how clearly they lie and misrepresent the evidence. Right? So one of the clearest examples is Haeckel's embryos, which some of you might be familiar with. But even the other evidences, they often would exaggerate or stretch the truth, or even outright lie. And so his first book is Icons of Evolution. The other one is Zombie Science, I think, which was only written about 2 years ago. And what he shows in Zombie Science is even after they are presented with, they are shown that you're lying, they still keep the same lies in the textbooks. And they add some new lies onto it. I think I've ran way over time, so I'll stop here. Subhanakallahumma wa bihamdika ashhadu an la ilaha illa anta astaghfiruka wa atubu ilayk.